Science Stand! Today we'll be talking about your research paper, a comprehensive molecular phylogeny of the Brachyurian crab superfamily Xanthoidea, provides novel insights into its systematics and evolutionary history. So, can you introduce yourselves, first of all?
Hello, I'm Dr. Jose Mendoza. I'm a carcinologist, that's a scientist who studies crabs. And I'm currently the curator of Crustacea here at the Lee Kong Chian Natural History Museum. Hello.
Hi, I'm Dr. Chan Kin Onn. I'm actually a curator of herpetology, study of amphibians and reptiles, not crabs. And I also work at the Lee Kong Chian Natural History Museum.
I noticed you're matching today.
Yes,
Was it coordinated?
No, totally unplanned. But because we are already in tune in the mind.
Same wavelength. Yeah. So why are xanthid crabs problematic or difficult to classify? Well, we have a handful of examples here in front of us. They're part of our museum's collection, large collection of Brachyurian crabs, right? Xanthids or xanthoids, they're a very big group and ubiquitous group of crabs. They're found all over the world, and they occupy a lot of different environments. They have different environmental niches, and because of that, they have evolved into displaying various and varied morphologies of forms. A lot of the classification in the past has been based on what a crab looks like. We rely on the shape of the body, the shape of the legs, etc., etc. This morphology-based classification system is wrought with complications. Sometimes totally unrelated organisms can be found in the same environment, and because of the conditions in that environment, they can evolve to have the same kind of form. Now, the job of the scientist is to find out what these real evolutionary relationships are. Here's the tree that came out in our paper. What this tree basically shows us is that the superfamily can be split up into 6 families now. Okay, traditionally, Xanthoidea has been broken up into just 3 families. So now what we're looking at here is a tree, a phylogenetic tree that is derived from DNA sequences of these crabs.
For this one, we focus particularly on using just DNA sequences. And then from this, there will be a lot of other follow-up studies that can focus on morphology, that can slowly get into the nitty-gritty of all these different subgroups.
Talk about markers in DNA. How do you extract the markers, or what do you look at?
Very good question. So you don't actually extract the markers. What you extract is DNA in general. So you try to get as much good quality DNA as possible. And then what you do is you design primers. Primers are like baits that target specific parts of the genome. You design a primer that specifically identifies that region of the mitogenome. And then through some lab work, you can get those markers to bind onto the specific region of the mitochondrial genome. And then from there, you amplify it, the specific part, and then you sequence it. So in essence, you are just picking up certain regions of the genome that you're interested in studying.
Any of the crabs that you extracted DNA from here?
Yes, as a matter of fact, this crab provided a important tissue sample that was used for the analysis in this paper. By the way, this crab is a highly toxic species of xanthid crab. From the name, the scientific name is Dimania toxica. Okay, and this is, I think, if I'm not mistaken, the first species of crab that was documented to have caused human death. Trivia for you.
Cool. Are you rolling now? Oh, sorry.
Okay.
Yeah, so I would say the first significance is that, first of all, the morphology-based classification does not 100% match what we see in the molecular-based classification system. So there needs to be some work to be done. And it's not necessarily to say that the molecular results is correct and is better, but it just shows that there's a lot of discord between these two different conclusions and that we need to look further into it. And I guess following up from that, the second most significant finding is that we understand so little about this big superfamily of crabs, right? They are global distribution, right? So globally distributed, more than 300 plus species, and our phylogeny shows that there could be a lot more species that have not been described. So that is also very significant. All of this requires a lot of basic foundation. If we cannot even put a name to a crab, then we cannot even proceed to study other things about the crab.
So this is what you two do every day at the Lee Kong Chian Natural History Museum?
I think this is what we would like to do every day.
Yeah.
But it's not necessarily what we do every day. Many different things that take out our time. And well, actually, JC mentioned earlier on a very interesting thing that, a lot of the tissues were collected by another colleague. So this is actually a very large and complicated project that involved many people and many institutions all around the world. And everyone was involved in doing different aspects of it. So this was a multinational, multi-year, multi-generational project, because I think it started way from our old boss, right?
Would you like to come to Singapore with us?
Professor Peter. So passed down through the generations, and now it's expanded through different institutions across all the different parts of the world.
All for crabs.
All for the love of crabs.